So I posted the solutions to homework 4 just now, and I'll talk through them a little bit today. There's one or two things I want to say. Let's see. We'll do that in a second. Last time we started talking about genome-wide association studies, and we talked about this idea of a single nucleotide polymorphism, a SNP, as a single location in the genome where there is known to be fairly, reasonably common variation in the population. We distinguished between mutations and SNPs, with really it just being kind of a convention of whether you call one a mutation or a SNP. Mutation usually is reserved for very rare changes, and often those rare changes are associated with, well, I guess that's not true. Never mind. But rare. Okay. And one of the relevant stories to the idea of SNPs and locating places in the genome where there's common variation is that gives you a series of what you could call markers along the genome. And we talked about how the vast majority of my sequence would be the exact same as your sequence. So why look at that? If I wanted to know the difference between me and you in a health way, say, no need to look at those. Instead, let's look at our markers, our some number of hundreds of thousands or millions of single nucleotide polymorphism locations where we know there is variation. And so if you have that, let's say we have 1 million SNP markers in a human. Those are all, right, and suppose that some of those SNPs can be associated. We're talking about doing association. GWAS, genome-wide association, is trying to associate SNPs with outcomes, like disease status, phenotypes. So can we find, in a particular disease context, say, can we find a marker or a series of markers whose status for an individual, genotype, is predictive of risk, say. So that kind of falls under the umbrella of personalized medicine, kind of. You know, if we can know something genetically that speaks to your predisposition to something, how you are likely to react to a medication, we could, for example, genotype a person on their 1 million SNPs, look at them, compare to guidelines based on published research, and say, this person will react best to dosage X of drug such and such. Personalizing the treatment to an individual based on their genetics. There are some services that are kind of interesting. Anybody heard of 23andMe? Yeah. So there are, given these newfangled technologies that can sequence lots of stuff quickly, relatively cheaply, you can get some interesting stuff about yourself. Yeah, I did it. So if you pay these people $100, they will send you an envelope with a little plastic container in it, and what you have to do is spit in it. And then they extract your DNA from your saliva.
You have to freeze it when you send it?
Nope. It may be a special kind of plastic or something, I don't know. So anyway, I thought I'd share some of the fun that you can see. Let's see, under my results, let's look at health risks. based on my markers, they can tell you things. We know stuff about Crohn's disease based on whether you have this SNP at this location or that SNP, what your genotypes are at these markers. They can compare those to known cases. Yes.
Is it whole genome sequencing or is just?
No, it's not the entire. They did not sequence my entire genome. They sequence only at these SNPs. I can't remember how many there are. So, for example, I'm at greater risk for Crohn's disease than the average population. I have almost 5 times increased risk of Crohn's disease. It's interesting that, well, anyway, I won't go through it in very much detail, but there's that. And then the kind of fun piece was this one. I don't know, maybe one of you guys know why a major category in the results is Neanderthal relation. How much Neanderthal composition is in you. Where did that go? Oh, Neanderthal ancestry. I'm in the 98th percentile. So, I don't know what that means. I have a heavy eyebrow ridge, long, low, bigger skull, prominent nose with developed nasal chambers for cold air protection. Doesn't sound like me, but anyway, it's kind of interesting. Okay, so let's look at the homework solutions. There's not really much to talk about with the first one because it's all code. My code is included at the back, and I will show you the picture that I had. This was my picture. You didn't have to have the pink. I just overlaid some pink regions to highlight where the actual genes were. But those were the coverages across the genome. I'm going to have Yabu take a kind of a quick look at your code, make sure it looks reasonable, and look at your picture, make sure it looks reasonable. It's not going to look exactly like mine. So full points if it looks, reasonable. I did have one read that misaligned. Didn't happen very frequently, but this was a read of 10 bases that, with high confidence, small p-value, was placed in the wrong spot. And the other, where did that go? Okay, yeah, that's it. So that's number 1. Number 2 had something that I wanted to comment on. So these were RNA-seq data, and you did clustering and PCA on the transformed read counts. Figure 3 is clustering results. So let's look at that for a second. And I specifically wanted to comment on the principal components. Here's the clustering, hierarchical clustering. And looking at the scree plot, we're looking for an elbow. And you could say you could argue for an elbow location in 2 places, which would result in your saying there's either 2 or 4 clusters. I think you could argue reasonably that there's 2 because of this very distinct jump, and to a lesser extent 4 because of this fairly distinct jump. I tend to be, I think conservative interpretation is a good idea when we're looking at clustering. Don't try to overinterpret. So for my purposes, I'm going to say there's 2. but full points if you said 4, as long as you said because such and such. If you say 2, then this last merge event is the one that I'm going to treat as truly distinguishing sources of structure. And what is obvious from what happened here is that we distinguished the control and the treatment groups. So the big difference between control and treatment. If you look a little more closely, this kind of mild evidence of 4 clusters would translate to once control and treatment have been separated, we can separate out time by time. So 2 weeks versus 2 months, and 2 weeks versus 2 months. Which makes sense that it would, I mean, it seems reasonable that there would be that kind of structure in the data. Clustering. Okay, then PCA. All right, so I could have been clearer on the task with PCA. The problem in the homework said, do principal components to explore structure among the rats, which are the columns. Remember what PCA is. It is get a covariance matrix and do eigenvalue, eigenvector decomposition of it. So if we have a data matrix capital X, that's M by N, traditionally, the way we think of capital X is that it has its variables in the columns and its observations or samples or individuals in the rows. So if you go to a multivariate statistics book and you look up principal components, that's what it's going to say. Suppose you have data, observations in the rows, variables in the columns, and you want to explore the structure among in the data. If you were to do, say, PR comp just on capital X, what it is going to do is treat it like it's set up the way I just described, as in individuals, variables. And we've talked a little bit about this, that's not really, we wouldn't typically think about our columns in our case as being variables. They're individuals. Those are the individuals. That said, PCA is perfectly valid as an exploratory technique. Either way, I could apply it to look for structure among the columns. I could also apply it to look for structure among the rows. And the way I would do that, so I could compute a sample covariance matrix that is I could compute a covariance matrix that by default, if I were to just compute a covariance matrix from this thing by just saying var x, then I'm going to have an n by n covariance matrix. Let's go to R and read in the data so we can follow along here. Scripts. So my code is here in the solutions. Let me copy this out. How do I want to select text. I need to change my grabber to a cursor, basically. Thanks. So let's take this over to a text editor. Come on. So the spacing got messed up, but that's okay. All right, let's read in the data. Currently in scripts. I want to go back in GS. Hammer. It's just hammer. Okay. Okay, let's see if that works. Hammer count table dot text. Hammer count table dot text. So I'm in scripts right now. I want to go back, and then I need to go into data. Okay. So 30,000 rows, 8 columns. Many of the rows have 0 counts in them, so we're going to get rid of those. And that leaves us with 18,600 rows. Okay, let's come down to the PCA section. So I'm going to library in DESeq2. Do we have that, please? Damn it. Packages. Okay, so I need BioC Lite. Okay, once we get that loaded in, we need to do the R log transform. And then here's the PCA stuff. So I have code in my solutions to do PCA both to explore the columns and to explore the rows. There were questions about what did I mean. I'm sorry, go ahead.
When we construct the EC2 object, we tell the R, the model, like the regression model that we want it to be to.
Mm-hmm.
Does that affect the R log and anything else that will apply to that?
The R log thing. Good question. Right. I don't actually know. That's a good question. was, as I understand it, up here we created a DE-seq object, and when we did that, part of what we were saying was this is the model we have in mind to use in a moment, and then we passed that DE-seq object into R log. So let's look up the help file for R log, because I'm not sure whether that incorporates the design or not. It makes sense to incorporate the design. If you're going to try to transform or normalize the data, it makes sense to incorporate what you know about where you expect differences to be. Normalization or transformation is often interested in trying to remove technical variation that's not due to the biology. So if you do know where the biology should be, it should help. I wouldn't be surprised if RLog does do something with the design, but I'm not certain. See if we can get this going, and no, not writable. Once we do this, once we have the transform data, two ways of doing PCA. So both cases I used PRComp. First time I transposed. Second time I did not. So when you do RLog transform, you can access the resulting matrix of new numbers by saying assay on the RLog output. So assay of YRLD is a M by N matrix of now continuous-like numbers instead of counts. When we transpose this, we now have an N by M matrix, and PRComp is going to interpret that as variables in the columns. So I have thousands of variables now and 8 individuals. Okay, so let's go ahead and set up DE-seq and do the RLog transformation. Probably to figure out what RLog is doing, we would need to go look at the paper. Transforms count data in a way which minimizes differences between samples for rows with small counts. Transform, minimize differences, okay, which normalizes with respect to the library size. Similar variance stabilizing effect, size factors. Yeah, that does not sound like it's doing anything with the model itself. It's basically just log transform, but it's concerned with the fact that in these NGS data, there's often a lot of zeros. So if you log transform zeros or numbers that are really close to zero, you get computational issues. I think that's what it's doing. Let's do PCA. When we do this, PCA, if I transpose the assay, if I print PCA right now, I'm going to regret it because it printed everything. It printed way too much. Why it did that? Why did it do that? Because when we transposed X, we had, so X transpose was N by M. That's going to give me a covariance matrix that's M by M. So 19,000 by 19,000. And then PCA is going to find linear combinations of the columns now, the M genes, linear combinations of the genes that explain the most variance. So if I were to try to look at the PCA output, what it usually tells us is here are the variances of the principal components, and here are the coefficients that define their linear combinations. So there's going to be 18,000 linear coefficients for PC1. So the question that I asked in the homework, interpret the coefficients, is not very straightforward here. You can't visually interpret 19,000 numbers. Here we can. If I don't transpose, then I'm going to have an n by n, 8 by 8 covariance matrix, and the first PC is going to be a linear combination of the 8 individuals. So there's 8 numbers, which we can look at. So just be aware that it depends. What PCA is doing depends on whether you, how you orient your matrix. They will tell you different things. They're both useful to do. Here is the output from doing PCA to explore let's see. So if we do this, we're going to be exploring. So we did log transform, R-log transform. That's a object of a special class. And in particular, I can't access the numbers that are in there. If I want the numbers, I can pull them out with assay. So this is the same dimension as our original matrix of read counts, but we've turned the integer counts into continuous-like numbers. Assay. So now those aren't integers anymore.
Gives you the same results.
If you don't use assay, it gives you the same results. PCA works? Okay. So it must know how to, if you try to do math with this object, it must have methods that know how to interpret that. But if you want to actually look at the numbers, you can pull them out with calling assay. So apparently that's not strictly necessary in this case. This that we just did here, okay, so let's look at some of the output. Percent explained. Inside PCA, the output of PCA we've seen before, or PR comp, if I say names PCA, there's the standard deviation, there's rotation, and the rotation is the thing that has 18,000 rows in it. Let's look at that. Standard deviation. If I square those are the variances of the principal components. The sum of the variances of the principal components equals the sum of the variances on the original data. So the proportion of variance explained, these are the eigenvalues. Those numbers are the eigenvalues. Yep. These are the proportions of variance explained. So 75% explained by the first principal component, 11.9% explained by the second. I can't look at the coefficients of those linear combinations, so I can't, we could try to dig in and do that. We could make pictures and stuff like that, but it's not easy to look at 18,000 numbers and say, what does the first principal component represent? It is easy to figure out what PC1 represents by looking at the picture. So we're not really in a bad spot. I just can't look at those coefficients. First PC explains 75%, second almost 12%. What I would do next is make a picture. So let's go ahead.
How is this different than just using summary?
Could do that. I think if I say summary, okay, that's not bad. I just didn't want to fill my screen with printout.
This is where our numbers are different.
Oh, you don't have,
It says my first PCA was 99.197.
so that's what happens. Did you transpose here?
No, I didn't.
Okay, so then you have the right answer for that application. We're going to do that here. So what we're showing is that you can do PCA. When you did this, you didn't use assay, which is fine. When you did this, that corresponds to take the original data, get its n by n, 8 by 8 covariance matrix, and then the 8 eigenvalues and eigenvectors of that. 99.5% is explained, we'll see, by that first principal component. Only 75% of the variance is explained by the first principal component if you look at it differently. So I'll revisit that in a second. But roughly speaking, what this is going to mean for us is there's a different story in terms of what separates the rows from each other and what separates the columns from each other. So let's keep going through this, and I hopefully will fill in the blanks. Up at the top, when we transposed the data first, my next step would be to make a picture of the principal components. Here we have 18,000 dimensional data. So there is no 18,000 dimensional picture that we can make. But the good thing about principal components is that you have the ability to bring it from 18,000 down to, say, 2 dimensions in the most meaningful way, in the way that preserves the structure as much as possible. So what we could do is make a 2-dimensional picture using these principal components. Let's make a picture, a scatter plot of the first 2 principal components. That's what this is doing. And it looks like this. I'm going to put the other one right here in a second. So PC1 is horizontal direction. That's the dominant direction. So differences horizontally are more impressive, I guess. They're more they're driving things more than changes vertically, because the vertical direction only explains 12%. So if we look at the horizontal picture or orientation, there's a pretty clear pattern. There's 4 points, very much separated from these 4 points, horizontally. The horizontal direction is how you're separating. Yes?
Here we are looking for a structure in the rows, in the observation.
We are looking at how the individuals, we're looking at the structure among the individuals, the columns. PCA, if you give it a rectangular matrix, will tell you about structure among the columns. The variables, classically we think of the columns as containing the variables. In this case, the columns are our assays, the individuals, the RNA-seq samples. So I have 1 point for every column. And I can look at this picture, and any apparent groupings or clusterings or outliers or anything like that are groupings and clusters and outliers, roughly speaking, in the full dimensional space, because we boiled it down in a way that we still preserve, 86, 87% of the total variation. So I would look at this with just 8 points, and I know that the horizontal direction is the biggest place to look for explanations, and there are 4 points that are very clearly distinguished from the others horizontally. Yes? About PCS? P- oh, yes, sorry. Yes. So what we just made a picture of is the actual principal components themselves. And a principal component is defined as if X is a vector that contains some number of variables. So suppose X is 8 numbers. Let's not say 8. Let's say 18,000 numbers. It is a column. That's for the second one. Let me go back to what I said originally. Let's say X is 8 numbers. It is a row of the original data set. In those 8 numbers are our 8 individuals, and for a moment we're going to consider those as variables. A principal component is going to linearly combine those variables. So give me 8 numbers, and I'll do E, the first one, times the first column, plus the second one times the second column, plus ... This would be a principal component. And if this was the first eigenvector, if E was the first eigenvector of S, this would be the first principal component. We could call it Y1 or PC1. In the code, what we did was we have X. We have X, and we also have, let's call it E, where E is 8 by 8, and it contains first eigenvector in the first column, second eigenvector in the second column, 8th eigenvector in the 8th column. So if I want, say, the first principal component, what I need to do is matrix multiply X with E1. X is m by n, E1 is n by 1. So this is going to turn into an m by 1. And so for the ith entry, it's going to be PC1 evaluated on the ith gene. So in the code, we did basically X matrix multiplied by E. And that gave us, in one fell swoop, the m by 8 matrix. So the first principal component is in the first column of that, second is in the second, and so on. That's what happened with PCs. And then in the plot, I said just take the first 2 columns. That means what that will do is plot the first column against the second column. Does that make sense? I think it does exactly the same thing, plot PCA. If you just do plot PCA, I have it down here, it's going to not do the transpose. It's going to basically assume, it's going to give you these percentages that you saw whenever you did not transpose. So if you just call plot PCA on, go ahead. Oh, the int group thing? Yeah. You didn't used to, I don't think.
I'll try to run it. I'll see what happens. But it should be doing the same thing. It's just PCA
Just the color uses the groups.
to color.
Color differently. So
Okay. All right, we'll do this in a second. We finished the story here. When we made our picture, there's these 4 points, those 4 points. Let's try to figure out what the story is. Apparently, there is structure. 75% of the whole picture can be summarized with this horizontal pattern. And there's clearly 4 versus 4. So how could I figure out a explanation? One thing that we could do, the principal components themselves, it's an 8 by 8 matrix. Let's look at the first principal component. Okay, those are the actual numbers. And in there, we have the column names. So the first, the value of the first principal component for sample 1 is minus 32. That for sample 2 is minus 31. And then for sample 3, it's 13. Sample 4, it's 16, and so on. So when we look at this picture here, negative 30-ish, negative 30-ish, 14-ish, 14-ish. And if you look at these numbers, the big negative numbers are all control group. And the positive numbers are all treatment group. So by applying PCA to, quote, explore structure among the individuals, the columns, we see that the columns do separate out. There is some systematic difference between columns. And in particular, it's to the tune of 75%, and it can be interpreted as a control versus treatment difference. So this picture right here is telling us something very similar as what the clustering picture told us. Now let's look at our data from a different angle, which is to say, that's how the columns relate to each other. Now let's look at how the rows relate to each other. So columns relate to each other. What does that mean? That means each column is 18,000 genes, read counts for genes. So each column is a vector, big multidimensional thing. And we are looking for, well, we're looking for linear combinations that vary a lot. And apparently, contrasting the treatment groups is the most informative way to combine the columns. If there was no difference between the treatment groups, then we would see something very different from this. So here's an exploratory technique that says there's good evidence of differences between these groups. PC2, I forgot to look at that. Let's look at PC2 real quick. You could probably guess what it's going to be. I'm less interested in it because it only explains 12%. So even though that point, in terms of how much space my eye has to go, that point is far away from that point. This point is further away from that point in some sense, because this is a more informative direction. So just, I don't know if that made sense or not, but vertical direction, let's look at it. There is some separation vertically. Here's 2 points, there's 2 points. Here's 2 points, there's 2 points. Ignoring the horizontal, there's 4 points up here, and there's 4 points down there. So we can separate our columns this way, and then independent of that, that way. So what is this direction doing? Let's look at PC2. They're all of the same sign. They're all negative. How would you interpret that? What does PC2 capturing or highlighting. If you were to apply that linear combination to a gene has 8 numbers for you, read counts. If you did minus 74 times the read count for control 2 months, plus minus 68 times control 2 months, second observation, plus ... how is that going to be extreme? How is that... They are the same sign, so we're not exactly contrasting.
I think there's still some, there is about 10% variation that we need to adjust for. And this mean, it's a constant coefficient of the, actually the mean, it's like the mean of all the columns.
Yeah.
We still need this. So if you plot them, you see that we cannot cluster all the samples, all the columns,
Yeah.
just the first one.
With just the first one. So this is a supplemental,
Yeah.
a little more subtle pattern that is still, fairly substantial, 12%, and it is necessary to completely represent the data. I had an explanation for this last. Huh? It is months by weeks, and I'm just having trouble seeing it. The first principal component was picking up on control versus treatment. Second principal component is picking up on time differences. So how is that? So if this is picking up on control treatment differences, PC1 is going to kind of blow up when I get big in magnitude whenever we come to a gene that differs between control and treatment. You got maybe small read counts in control, big read counts in treatment, so this linear combination is going to highlight that gene. Down here, go ahead.
The second ones are about 10% larger than the second 4 values.
About 10%, yeah.
They are about 90%, but the first 4 are about 70, 75.
Yeah. Yeah, so the last 4 are different from the first 4. These are all minus 88. These are all minus 70-ish.
That's the time variable.
That's time. This is 2 months. This is 2 weeks. So that is what it's doing. I'm not explaining it real well. First PC is pretty easy to explain, differences in control and treatment. Second PC is more subtle. I guess the punchline at this point is that we have done PCA of one type to these data. We are looking for how do the columns vary in structure with respect to each other. It's possible that you have, in this case we do, differing levels of structure depending on whether you ask about columns or rows. All right, let's look at the rows real quick. And PCA, PR comp, assay. Okay, now we only have, here's the output of PCA, or PR comp. We have our eigenvectors. There's only 8 numbers in the eigenvectors now because we have transposed the data. If I transpose, then, let's see, no, I have not transposed. We did not transpose this time. So we're dealing with 8 by 8 covariance matrix. The story is a little different now. The first principal component explains almost all of the variation in the data, 99.8%. And we've seen this kind of pattern before, several times now. How do I interpret the first principal component? I could, in this case, just look at the coefficients. And they're all the same sign, virtually the same magnitude, which we have said is indicative of strong correlation. All of these variables, are strongly correlated with each other and have similar variance. So they all kind of equally contribute, and you need them all to represent the whole. The most informative way to linearly combine them all is to average them. Second principal component, I wouldn't bother even trying to interpret. It does not explain enough to really put much faith in. I can do the same picture that we made above. Let's compute the principal components. So now I take my capital X matrix and multiply by the eigenvectors, but I'm not transposing this time. And then here is the scatter plot. So it looks very different. Now I have 18,000 points on there. And I don't see, in this case, obvious grouping or clustering. You know, I don't see a big blob in the upper right and a big blob in the lower left, for example. They're just all kind of smeared together. The beak? This thing? Yeah. It's not always easy to explain kind of weird patterns to data. They're not uncommon. Weird patterns like this tapering off to a point. So that's happening. PC2 is going to 0 over here. I'd have to think about it to explain why that is there.
Seems like those genes are pulling off from the structure.
Yeah, maybe you could think of these as distinct somehow. You know, they're low.
Because that's how I thought it was. I was like, well, maybe these are using different expressions compared to everything else. Subtle. Pulling it off.
That could be. In order to dig a little deeper, I would want to look at the actual principal component numbers. So I would look at PC1 numbers and try to isolate maybe some of these that are extreme over here and see if anything jumps out about them. You know, like look at the actual numbers, the actual raw data for these guys. In fact, let's do that real quick. The ones over in the beak are the lowest PC1 values. So PC1, let's order PC1, order PCs. The first one, decreasing, is false. So order from least to greatest. Then if I do that, let's just get the first 50. The 50, no, I don't want to do that. I want to look at the raw data. Let's look at the raw read counts for the 50 genes that are furthest over here. Just curious. I don't want that, I want this. So that's why. They just barely didn't get thrown away. Each of these had one. So that would tell me these kind of tapering and odd patterns to a scatter plot often have to do with kind of extreme values in your data, either extreme small or extreme big. Often extreme small, like there's a lower bound. I can't have a negative read count, but I have lots of zeros.
But still, I mean, they're expressing for one scenario.
Yeah, that's true.
Treatments.
Yeah. I'm going to end right here, but I will make a comment that these kinds of genes right here, there's not much we can do statistically using what we know. Like a t-test, for example. Try to do a t-test on data where there's 1 1 and 7 zeros. You can't compute a variance. It's not reliable. But suppose you did have data like this.
It's only one rep though, so that makes.
Yeah, it's just one rep. So it could be, maybe there's something weird about that rep. But you could imagine cases where you are specifically interested in not testing is the mean difference between treatment and control, say, but is presence absence different? In which case, the ones and zeros are natural things to work with. All right, anyway, I think the beak is kind of an unnatural consequence of these really low read counts for these genes. Overall, I don't see grouping like we did over here. The big smear is kind of what you would expect from data that are really highly correlated, like these. Really highly correlated, 99%, 87%, 97%. That's the correlation matrix. Okay. Questions? We've spent a lot of time on PCA. I hope that you're getting good things out of it. It's a good technique. Clustering's a good technique. They both do kind of similar things, but they're operating differently, so they can tell you slightly different stories. I would recommend always do both, because one may tell you one thing, the other may tell you something a little different. Okay, that's all I got to say. All right, then I will see you on Monday. Have a good weekend.